In 2005, a forensic geneticist in Copenhagen wasn't trying to rewrite ancient history. He was building a DNA database to identify crime victims. But buried in his data was a number nobody in Egyptology ever saw: 77.6%. That's how often one genetic marker showed up in 200 Somali men. A frequency so high it only has one explanation: that lineage has been sitting in the Horn of Africa since deep prehistory. The paper got published. Then it sat ignored for 19 years. Not buried by conspiracy, buried by bureaucracy. Forensic geneticists in Denmark don't publish in Egyptology journals, and Egyptologists don't read forensic crime databases. Meanwhile, the ancient Egyptians themselves had been telling us where they came from the whole time, a place they called Punt, the land of the gods. Modern scholars filed it under myth. Turns out the DNA agrees with the Egyptians, not the textbooks. Here is the question this video is going to answer, and I want you to hold it in your mind for the next 9 minutes. How does a forensic genetics paper, commissioned for a Scandinavian court system to identify crime victims, end up containing the single most significant data point in the debate over who founded ancient Egyptian civilization? To answer that, we need to understand what haplogroup E3b1 actually is, because the Sanchez paper wasn't just recording a statistical anomaly. It was, without knowing it, measuring the depth of something. In population genetics, when a Y chromosome haplogroup exceeds 50% in a non-isolated population, one of only 3 explanations holds: an extreme founder effect, intense natural selection pressure, or deep ancestral origin, meaning this lineage has been present in this population for a very, very long time. Somali populations are not genetically isolated. Trade routes have crossed the Gulf of Aden for 5,000 years. Multiple distinct genetic waves have entered the region: West Eurasian backflow, Arabian admixture, Indian Ocean commerce. And yet E3b1 sits at 77.6%. That is not genetic drift. That is a signal from deep time. And here is where the forensic database and the ancient world collide. Because that same haplogroup E3b1, now classified as E1b1b, appears in something else entirely. It appears in pre-dynastic Nile Valley burial sites, at its highest recorded frequency, before Levantine Near Eastern admixture arrived, before the Pyramid Age, before unification under Narmer, before Egypt was Egypt. Let's build the case properly, because this is where most coverage of this topic gets lazy. The Egyptian texts. For over 2,000 years, ancient Egypt described a place called Punt, Tanecheru, the land of the gods. Not a trading partner, not a colony. Their own inscriptions describe it as the geographic origin of the Egyptian people themselves. Queen Hatshepsut's 18th dynasty expedition, around 1470 BCE, recorded it in stone at Deir el-Bahari, the most detailed account we have. Tall figures, braided hair, distinctive cattle. Frankincense trees carried back to Egypt with their roots still in the soil. For 150 years, Egyptologists argued about where Punt actually was: Sudan, Eritrea, Yemen. Some argued it was mythological, a spiritual concept, not a real place. Then in 2020, Dr. Nathaniel Dominy at Dartmouth College ended that argument with the teeth of dead baboons. The ancient Egyptians mummified baboons as sacred offerings to the god Thoth. Dominy's team ran strontium isotope analysis on the tooth enamel of those mummified animals, comparing their chemical signatures against 77 geographic locations across the African continent. Strontium is absorbed from local bedrock and water as an animal grows. It is a geographic fingerprint written in calcium. The match was unambiguous. Those baboons, holy enough to be wrapped and placed in Egypt's most sacred temples, came from the Horn of Africa, from the coastal corridor of modern Somalia and Eritrea. Punt was real. Punt was in the Horn. And Egypt said Punt was where they came from. The genomic data. In 2019, Dr. Mary Prendergast at St. Louis University analyzed genome-wide data from 41 ancient individuals associated with the pastoral Neolithic in East Africa. Her conclusion, published in Science, was precise. The genetic signal shared between Nile Valley populations and Horn of Africa populations does not represent migration from one to the other. It represents a common ancestral source, a single founding population, whole and undivided, that both ancient Egypt and the Horn of Africa descended from. And here's the critical asymmetry that changes everything. After Egypt's founding period, the Nile Valley absorbed successive waves of Levantine Near Eastern farming ancestry, a well-documented admixture process beginning around 3100 BCE with political unification. The Horn of Africa did not receive those waves in the same way, which means the Horn preserved the original ancestral signal at higher fidelity than Egypt did. The less diluted form of the common ancestor is not in Cairo, it is in the Horn. Now, bring the forensic database back. The Sanchez 2005 paper was cited 89 times between publication and 2020, almost entirely in forensic genetics databases and population identification literature. It was cited 0 times in any academic paper that used the phrase pre-dynastic Egypt or pyramid builders in its title. 0. Not because the data was wrong, not because the methodology was flawed, but because a forensic geneticist in Copenhagen and an Egyptologist in Oxford read different journals. And nobody, for 19 years, sat at the intersection long enough to ask the obvious question. The highest measured frequency of the founding Y chromosome haplogroup of pre-dynastic Egyptian civilization anywhere on Earth, sitting in a crime victim identification database, and the field of ancient history never looked. That is not suppression. That is what happens when disciplines don't read each other's journals. But the effect is identical. Knowledge, stranded between fields, waiting for the right question to find it. 100 kilometers east of Hargeisa in Somaliland, inside a cave complex called Las Gil, are rock paintings dated between 5,000 and 11,000 years old, among the oldest in Africa. Domesticated cattle with elaborate ceremonial markings, human figures in sophisticated dress, cultural complexity, codified in pigment, thousands of years before the Egyptian Old Kingdom. Beneath those paintings, human skeletal remains. Material dated precisely to the pre-dynastic window, 5000 to 9000 BCE, the exact period that has never been genomically sampled. The founding window, the one that would answer, with hard evidence, whether the Horn preceded the Nile or simply shared the same ancestor. The ancient DNA technology exists. The skeletal material exists. The funding does not. No major ancient DNA consortium has allocated resources to extract ancient genomic data from Las Gil. The site that could close the most consequential open question in African population genetics is sitting in the Horn of Africa, undisturbed, because the field has not yet decided the Horn is worth asking. Dr. Pontus Skoglund at the Francis Crick Institute, one of the leading figures in African ancient DNA research, described the Horn of Africa in a landmark 2017 Cell paper as containing a genetically distinct lineage of population history that remains, and I'm quoting directly here, significantly understudied relative to its likely historical importance. Significantly understudied relative to its likely historical importance. One of the field's own leading researchers said it plainly in 2017. 7 years later, the Las Gil remains have still not been sequenced. 9 million people carry the E3B1 lineage in the Horn of Africa today. Farmers, engineers, teachers, parents, living their lives with no awareness that their Y chromosome carries what may be the closest measurable biological echo of the people who conceived the earliest known civilization. The oldest surviving wonders of the ancient world have no idea they're still there. A forensic database, a mummified baboon's tooth, a cave in Somaliland with 9,000-year-old bones, and no extraction funding. 3 separate threads, one conclusion the data keeps pointing toward, and the field keeps failing to fully follow. In population genetics, when the direct ancestral sample is unavailable, researchers use the least admixed living population as the best available proxy for the founding ancestor. The population with the lowest subsequent Near Eastern admixture, the highest E3B1 preservation frequency, the closest geographic link to the Green Sahara dispersal zone. All 3 metrics point simultaneously to the same population. The counter-argument's strongest point is also the argument for why this story isn't finished. The arrow we drew on every history map for 150 years, Egypt as source, Horn as recipient, the genomic evidence, consistently, for the founding period, points it the other way. If ancient DNA found in ice can upend what we thought we knew about Bronze Age Europe, and it can, then ancient DNA extracted from a cave in Somaliland would do the same for the oldest chapter of African history. We've seen what happens when genetics meets a body that shouldn't exist in the historical record. If you haven't watched the Ötzi video yet, that's exactly where this kind of science leads. It's linked at the end of this video. I'd start there.